switch to lecture 17, and we are going to talk about extracellular matrix. Basically the fibrillar matrix or network that deposit in the outside of the cell, and definition, and then physical chemical properties. And then we'll just touch the the most abundant protein in the extracellular matrix, collagen type 1. Okay, so the organization of the cell and matrix, extracellular matrix, is the this is showing. I'm going to show you something like this many times, and that is the intestinal lining, and showing the highly organized tissue here. And also, you see a very loosely organized tissue is sandwiched by the those tightly organized tissues. So I would say here that is loosely organized connective tissue here. And then you have this muscles, okay? This is the muscle structures, okay? And I think that another loosely organized tissue here. So I think that this is not really epithelial, so this is the muscle. Okay, okay. And then this is the epithelium. So that is the real, epithelial tissue that has the strong cell-to-cell interaction. And then this is the epithelial cell, another cell. The other side of the, other side, okay, of the, intestine here. And this is also epithelium. So if we look at, isolate this area, this kind of like a, connective tissues that we are going to talk about. Mostly they are formed by this fibers. You see this fibers over here, right? So this is the extracellular matrix, proteins, okay? Mostly extracellular matrix proteins. And then you also see this, cells, okay? So cells are, mixed in there. And I think that's the cell surface is touching these fibers, okay? So these fibers are really touching the surface. So mixture of this kind of fibrous protein and then cell, that is, I would say, the connective tissue that we are going to talk about. Okay, so why this extracellular matrix is really important is that, okay, so not only, they are kind of like a, forming this kind of fibers or something, but actually they are important in this cell to ECM interaction. Or sometimes I would say cell to matrix interaction. Okay, right. So they are binding to, anchor to this extracellular matrix. So I'm sure that they have a particular receptor here. Okay, and does that. So cells like interaction is largely these two interaction. Cell to matrix. I think that is like a cell to almost like an environment. I would say microenvironment. And then this is showing here, and that is another type of interaction that is more important, cell to cell interaction. Okay, right. So, in relation to what I have just talked about in the previous slide, I think the cell-to-cell interaction is more predominant in epithelial cells. I mean, they do have this, cell matrix interaction, okay? But the cell matrix interaction is mostly, some kind of mesenchymal cell. Cells, or yeah, mesenchymal cells. I would say some kind of migrating cell, something like that, has the more this cell to matrix heavy proteins. Cells. Okay, so those cells. Okay, let me switch the color. So those extracellular matrix, mostly they are fibers. Okay, so they have collagens and elastins. I'm sure that they have more. And then ground substance is that glucosaminoglycans, the sugar, and the proteoglycans, okay, and glycoproteins, and then water, salt, and then other nutrients mixed together, and then they are forming mixture, and then forming this kind of matrix-like structures. So one of the tissue example is what? Let me read this caption here. This is a full thickness skin of newborn infants, showing the relative thickness of the epidermis. So here is the epidermis, highly organized, I guess. See? So due to the cell-to-cell interaction, okay? Interaction. Okay, and I think that they have some very, very, strong protein layer, okay? So that is the basement membrane. Okay, I think that we're gonna talk about this basement membrane, but that is also extracellular matrix, okay, ECM. Okay, right. And then from here to here is, I would say, dermis. So that is like a connective tissue. Okay, and then it is loosely, organized. Okay. All right. So what is this basement membrane? Basement membrane is that they say it's a membrane, but this is not a cell membrane. It's not a lipid bilayer. And then this is the protein layer, and it's a special extracellular matrix. So connective tissue ECM is the big thing, and then I think that the basement membrane, BM, is one of a kind, but pretty narrow. So it's like a, I draw this Venn diagram here to help you understand, extracellular matrix and connective tissues. Okay, so we are looking at those, dermis area in a little bit higher magnification. Okay, and as they go deeper, and then you can see, fibers are getting thicker. Isn't it? Right. So thicker, deep in dermis. And then you can see fibroblast in here. Honestly, I cannot see those fibroblasts. I think this thing looks like a fibroblast to me. And they have even label here. It's really hard to see if you follow those. Here is one label here. Isn't it? Somewhere they are showing this, fibroblast. So there's some cell that moving around, kind of like the forest of fibers, and then some kind of cells kind of swimming around. Okay. So here is mostly like a fibro, collagen-like, fibers, and then you will see more elastin or some elastic fibers are more, accumulated deeper in the tissues. Okay, so basically, I want to summarize is that there are two types of ECM, and one is the basal membrane and the connective tissue. And basal membrane is old name is a basal lamina. You're gonna hear about this basal lamina a lot because some old folks still use the basal lamina, including myself. And the connective tissue, that is interstitial matrix, those basement membrane is very sheet-like structure, act as a support for epithelial cell, and its permeability and everything. But I would say basically, where the tissue is, and then tissue boundary between tissue to the another tissue, you always find the basement membrane. I think that when you are taking anatomy or histology class, and when you look at those tissue sample, I think that's your instructor will ask you to identify basement membrane first, because that is the kind of that's demarcate the tissues, borders. So you will know where is the boundaries, and then from that you can trace through all those tissues. So that is the basement membrane, and then it's very distinct, like a thin line, okay, pretty thick. But connective tissue ECM is a network of fibers. It's amorphous, and then loosely organized, so it's very porous, okay? But if you look at it component-wise, in basement membrane, okay, I'm talking about basement membrane. They have these 4 components. There is predominant collagen type 4, perlecan. There is a proteoglycan, and then as a glycoprotein, laminin, and intactin do they have, okay? But in connective tissue, major components of this connective tissue ECM is type 1 collagen, and then decorin and other proteoglycan, and fibronectin, okay, as the glycoprotein. This is mostly like a vertebrate mammalian tissue, okay? Okay, so we are talking about, okay. So if you look at those kind of like a diagram of those ECM components, they are green is, I think, that's glycoprotein, and you see it's a connective tissue molecule, and then basement membrane molecules that you can see, and this is the tenascin. We did not talk about it, but this is connective tissue. And then I think there's proteoglycan has this red line here. So this is sugar, okay? So very, very big chain of linear sugar, okay? And then all this, proteoglycan, they have this sugar decoration in it. I think that's a major function of the this PG, proteoglycan, is coming from sugars, okay? I think that is one of the definition of this sugar residue in here. Okay, so I'm going to move on. So the features of those ECM proteins, if you look at it, and they are very big, and they are secreted, and they are glycosylated, and they have a disulfide bonds because holding the certain structure. And then they are composed of multiple domains and multiple subunits. Sometimes, alpha, beta, gamma, 2 protein comes together. And then because of that, they have a multiple functions, okay? I think that we are going to talk about what those mean, and then what kind of structural motif they have. We are going to talk about a little bit, 1 or 2 domains or motifs that is very important in here. Okay, so when you look at it, here this is the fibronectin, and type 3 fibronectin here, and they have this fibronectin type 3 repeat. I think they have about 15 of them in here, and then those same fibronectin type 3 repeat is repeated in this tenascin, and then, you know, integrin and other molecules, okay, right. And if you look at the, you know, other, you know, abundant one, this EGF repeat, the round one, as you can see, laminins, do they have an EGF repeat? Tenacin has it, and also agrin, and then also some of the other protein has those commonly found repeat. So fibronectin pathway repeat is, and then EGF repeat is the one of the most abundant domains in the extracellular matrix proteins. Fibronectin pathway repeat is almost same as Ig repeat, which is the immunoglobulin repeat. So, you know, it has a disulfide bond. This one has disulfide bond, but this one does not have disulfide bond. But basically, they are identical globular shaped structures. So we'll touch base the most abundant collagen, that is the 30% of body's protein. You know, we are using Jello. Gelatin is the denatured collagen, and I think that is we use it for food. Okay, so collagen type 1, okay, is also important feature is that they are forming this triple helix, so composed of the three you know, say subunits. Okay, I think that's, you know, alpha 2 and alpha 1 chain. I think that either alpha 1 or alpha 2 has the 2 subunit and then 1, and then they can form this triple helix. So the triple helical region, how they form triple helical region is that triple helical region, they have this kind of repeat, glycine, proline, and anything. Okay, just keep on going, glycine, okay, proline, and then X, and then glycine, proline, X. They'll just keep on going like this, repeat. And then if you have this kind of protein, 3 of them together, then you can form this triple helix here. So those triple helical, you know, protein bundles up. So this is one of their triple helix. Okay, so this is the triple helix, right? And then they will form this fiber, and then they have an end-to-end joining, and then also the cross-linking, of course. So they can form this, you know, bundle. Okay, right. So triple helix is talking about this. Triple helix is that this individual, just one bar here, that is the triple helix. Okay, and the this is like a 3 triple helical fibers comes together, and then forming this kind of and by their cross-linking and end-to-end joining, they are making this bundle, fiber bundle. Okay, so that is what is representing this in figure 5. It says collagen fiber, right? Okay. Right, so collagen fiber is very, you know, abundant. You take the tissue slice, and if you see it, you can see this is the fiber, and also these are all collagen fibers because they are crisscross. And then when you cut it, some fibers in an opposite direction, and you see cells over here and here. This is typical connective tissue, you know, environment. So collagens are actually, we talked about this number 1, but they have so many collagens. I think there's more than like 25 collagens too so far. So I don't want you to memorize this, but something that we are going to talk about is at number 1. And then I think that we're going to talk about this, and maybe we may talk about one of this later. But those are the ones that we are talking about. I think that's something that I circled. You probably want to you know about these you know proteins. So I'll finish the first part here, and then I'm going to move to next segment next time.